Clearly, ma'am.
Hello, is my voice audible? Hello. Yeah, sorry, I'm very sorry. There was a
Here's the
internet
current
issue.
one.
There was a internet issue today, so very sorry. Uh, because I have to leave and just let me restart the meeting. Okay. I'm really sorry for the disruption. Don't know why it is happening today. So is my screen visible? Just let me know if my screen is visible or not. Okay, so very sorry, there was an internet issue and there is frequent power cut is happening today here. So I'm really sorry. So I will just restart the session. Okay, if it is fine with all of you, I will just restart the session. Yeah, so very good evening to all the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for the NPTEL course of Organic Chemistry in Biology and Drug Development. So before starting the session, I would like to brief the few points, like there will be a problem solving session every week. These sessions will be on Tuesday and it will be from 6 p.m. to 8 p.m. The sessions will be live on the Google Meet platform. If any network disruption happens, please wait for a few minutes. Do not leave the session. Okay, I will join as soon as possible. So during the session, the students can post their queries and doubts through the chat box. If still any doubt persists after class, the students can drop an email to me. I will answer their query. Okay, this presentation and class recording, it will be shared to you soon from the NPTEL website. Okay, so I think I will just start today's session. I am already late. I am very sorry because there are frequent power cuts today. So please do not leave the class. I will just join as soon as possible. So I hope everything is fine with the PPT right now and also with my sound and voice. So let's start the session. Today it is the week 6 live session and today we will discuss the week 7 course content. So in the week 7 you have molecular biology and chemistry of cofactors and coenzymes. So all the questions which we will discuss today will be based on these two topics, molecular biology and the chemistry of the cofactors and coenzymes. So the first question of today's session is, for which restriction enzyme only manganese ions are essential? Your options are number A type 1, number B type 2, number C type 3 and number D all of the 3. So let me know for which restriction enzyme manganese ions are essential, type 1, type 2, type 3 or all of the 3. I would like to request all of you to try to answer the questions. It does not matter if your answer is right or wrong, but please try to answer every question, okay? Because that's how I will know that your if you have understood the topic or not. Anyways, we will discuss this thing in detail. So let me check if there is any answer in the chat box. So my voice is audible, right? You can hear my voice and you can see the PPT. Yeah, okay. So, okay, let us discuss that question, okay. So, the answer of the first question is option number B, type 2. So, type 2 restriction enzyme, they requires only magnesium ions for the restriction. So, there are 3 types of restriction endonucleases. Your options are, the 3 types are type 1, type 2 and type 3. So, for type 1 endonuclease, what happens that the enzyme structure consists of 3 different subunits. For type 2, the enzyme structure is much simpler, and for type 3, the enzyme structure consists of 2 different subunits. The type 1 they require ATP, magnesium 2 plus ion and S adenosyl methionine for restriction. The type 2 they require magnesium ions for the restriction only, and type 3 they require ATP and magnesium 2 plus and S adenosyl for the restriction. The type 1 restriction enzyme they recognize the specific sites within DNA but do not cut these sites. Okay. The type 2 they recognize specific sites within DNA and cut these sites, and type 3 they recognize specific sites within the DNA but do not cut these sites. And for type 1 they are not used in recombinant DNA technology. Type 2 restriction enzymes are used in recombinant DNA technology, and type 3 they are not used in recombinant DNA technology. Okay. So, this was all about your restriction enzymes. So, remember there are 3 types of restriction enzymes, type 1, type 2 and type 3, and there are some comparison between these 3. So, I would like to request all of you to remember these ions. Okay. So, again I am saying, if you have any doubt, any query when their class is going on, please let me know, okay, in the chat box. Since there are frequent power cuts and internet issues, so please bear with me today because there might be some disruption. Just do not leave the class. If any issue happens, I will rejoin, but you do not leave the class. So, alright, is it clear? Okay, I would move toward the next problem. Problem number 2 says, which of the following statements is false regarding restriction enzymes? Your options are: number A, these are endonucleases; number B, these are exonucleases; number C, this recognizes, okay, there is any problem. Hello, is it all right? Can you see the next question? Okay, so next problem is, I will repeat again. So, problem number 2 says, which of the following statements is false regarding restriction enzymes? Your options are: these are endonucleases; option 2, these are exonucleases; option C, these only recognizes palindromic sequences; and option D, they digest double strand DNA at specific sites. So, which of the following statements is false regarding the restriction enzymes? We just discussed it in the last question also. Try to answer the question in the chat box. There is a power cut issue today in my area, so please bear with me. Do not leave the class. Okay, I will keep joining if there is any problem. So, okay, try to answer this question. Which of the following statements is false regarding restriction enzyme? These are endonucleases, these are exonucleases, these only recognize palindromic sequences, and these digest double strand DNA at specific sites. So, is there any question, answer in the chat box? Number B. Okay, so I have one answer which says it is number B. Now, let me check. So, your correct answer is number B. These are exonucleases. So, it is the correct answer. Okay. So, now as I have mentioned in the last question also, restriction enzymes are endonucleases. So, they are also called restriction endonuclease. Okay. Restriction endonuclease are the family of enzymes that each cut DNA at a specific sequence within the DNA. At a specific sequence within the DNA, alternatively called restriction enzyme. A restriction endonuclease can cut the DNA when it recognizes a specific sequence. These specific sequences are called the restriction sites. So, what are restriction enzymes? They are endonucleases. They digest double strand DNA at a specific sites. The recognition site is palindrome. So, palindrome means it will be same from the forward and backward also. Okay. So, just like as you can see from in this first example AIUI. Okay. So, it is 5 prime and then it is AGCT and 3 prime, and if you read it from the backward, it is same for the next strand, means it is A, G, C and T. Okay. So, the sequence which are same from the forward and backward, these are called your palindrome syndrome. Like madam, this word, if you look at this word carefully, M, A, D, A and M, if you read this word from this side and this side also, you will see it is the same. Okay. So, if this is the same, it is called the palindrome sequence. Okay. Any word or anything or any sequence, it is called the palindrome sequence. Okay. It reads same on the both strands from 5 prime to 3 prime and also from 3 prime to 5 prime. The recognition sites are different size and unique sites. Okay. They cuts between the same nucleotides on the each strand. So, if like from the first example AIUI, so it cuts between the G and C. So, if the enzyme cuts in between a particular base pair in one strand, it will cut in the same base pair for the next strand also. Okay. And this base pair is specific for a specific restriction enzymes. Okay. So, this was the problem number 2. If you have any problem, any doubt, please let me know in the chat box. Okay. Now, moving toward the next problem. Problem number 3 says reverse transcriptase requires which one of the following converting a single strand RNA into double strand DNA. The options are number A, all 4 NTPs, option B, all 4 DNTPs, option C, an RNA template, and option D, all 4 DDNTPs. So, reverse transcriptase requires which one of the following converting single strand RNA into double strand DNA? Reverse transcriptase requires which one of the following for converting a single strand RNA into double strand DNA? Your options are all 4 NTPs, option B, all 4 DNTPs, option C, an RNA template, and option D, all 4 DDNTPs. Is there any answer in the chat box for question number 3? Not till now. Okay, fine. Okay, let us discuss that also. So, the correct answer for problem number 3 will be option B, all 4 DNTPs. Okay. So, what is reverse transcriptase? So, reverse transcriptase enzyme is an RNA dependent DNA polymerase, and therefore, it produces a DNA strand from a RNA template. It has no associated exonuclease activity. Okay. So, as you can see, there is a reverse transcriptase, or it is in short form, it can be written as RT or RTS. Okay. So, there is a single strand RNA template here. Okay. And from this single strand RNA template, there will be some dinucleotide triphosphates. Okay. DNTPs, the full form is dinucleotide triphosphates, and when this dinucleotide triphosphates will be there, they will be polymerized by this reverse transcriptase, okay. And this, from this RNA template, RNA DNA hybrid will be formed, okay. And from this hybrid, the RNase enzyme, it will convert it from a single strand DNA, and from this single strand DNA again some DNTPs will come, okay, and all 4 DNTPs will join and it will form a double strand DNA altogether, okay. So, that this is the role of the reverse transcription. So, what is the difference between the transcription and reverse transcription? It is also very important. So, transcription is the process of copying the genetic information stored in a DNA genome into a complementary strand of RNA. Reverse transcription refers to the reverse process of the normal transcription, in which the RNA template is copied to form a cDNA molecule in retrovirus. Okay. Encoding of a DNA genome into RNA is the role of transcription, and in the reverse transcription, encoding the RNA genome into cDNA or cyclic DNA. Okay. So, the transcription, it occurs in the cytoplasm of prokaryotes and nucleus in eukaryotes. The reverse transcription occurs in the cytoplasm of the host. RNA polymerase is involved in the transcription, and reverse transcriptase is involved in the reverse transcription. No primer is used by RNA polymerase. Lysyl tRNA acts as a primer for the reverse transcriptase. The transcription products of the transcriptions are used in the protein synthesis, and products of the reverse transcription is integrated into the host genome. So, this was the comparison between your transcription and reverse transcriptions. What are the similarities, what are the differences, these are mentioned here. So, remember, reverse transcriptase, it requires all 4 DNTPs or dinucleotide triphosphates to form a double strand DNA from a single strand RNA primer template, okay. So, now I will move toward the next problem. Problem number 4 says the palindrome sequence recognized by restriction enzyme HIND3 is option A AAGCTT, option B CTGCAG, option C GATATC, and option D GAATTC. So, which palindrome sequence is recognized by the restriction enzyme HIND3? Try to answer it in the chat box. Which of the following is correct? Which palindrome sequence will be recognized by restriction enzyme HIND3? Is it AAGCTT? Is it CTGCAG? Is it GATATC? Or is it GAATTC? I will request all of you to try to answer the questions. It does not matter if your answer is right or wrong, plus please try to answer the questions. So, is there any answer in the chat box? So, I think I have few answers. So, number B, number D, number A. So, I think I have 3 different answers for this question. Okay, let me check which of the following is the correct answer. So, see, it does not matter if your answer is wrong, but answering the questions is very important. So, it was the last question. So, the correct answer is number A, CAAGCTT. It is the palindrome syndrome which is recognized by the restriction enzyme HIND3. So, this is the table which shows where different restriction endonucleases or restriction enzymes cleave DNA. The abbreviation R represents the purines, okay, adenine and guanine. The pyrimidines are abbreviated as Y. The abbreviation W, it represents adenine and thymine also. Okay. So, what is the cleavage stand for E. coli? Okay, E. coli, the target sequence is this palindromic sequence GAA-TTC 5 prime to 3 prime. I have mentioned in the first question itself that I think in the second question, okay, that the restriction endonucleases, they recognize palindromic syndrome. Sorry, not palindromic syndrome, they recognize the palindromic sequence. Okay, and what is the palindromic sequence? It will be similar from the forward and backward for the 5 prime to 3 prime. Okay. So, as you can see for the this E. coli enzyme, the sequence is 5 prime GAA-TTC 3 prime. Okay, and if you read it backwards like 3 prime CTTAAG, it is the next sequence. Okay, and this E. coli, it cleaves actually in between G and A. So, if as you can see, if the cleavage happens in this sequence, so these type of cleavage sites will form. Okay. Eco RV, it is the recognizes the site which is GATATC. Okay, and if again you can read it backward, it will be same GATSTC. Okay, and it cleaves between A and T. Okay, it cleaves between A and T. HAE3, it is the sequence is which is recognized by this enzyme is 5 prime GGCC 3 prime. Okay. And it cleaves in between G and C. Okay, it cleaves between G and C. And HIND3, which is in your question, it cleaves in between A and A. Okay, and the palindromic sequence which is recognized by this enzyme is 5 prime AAGCTT 3 prime. Okay, this is the palindromic sequence which is recognized by this. And there is another endonuclease which is PPUMI, and it is as I have mentioned, there is the abbreviation which is mentioned. So, it cleaves between G and G, and by G it means the guanine. Okay. So, there are many other endonucleases. Okay, there are few here, and I have mentioned it also in the problem number 2. Here there is a few other endonucleases. Okay, like we have AUI3, we have some here there is some examples again, like there is AIUI. Okay, so this is also very E. coli. So, these are very important endonuclease. So, I would like to request, or I would suggest that you remember each and every endonuclease, the common ones morely like E. coli, Hindi, AIUI. So, these are very common endonucleases. So, remember what are the cleavage sites, means in between which base pairs they cleave, and also the palindromic sequence which are recognized by these enzymes. Okay. So, now I will move toward the next problem. So, problem number 5, it says that the first step in PCR technique deals with your options are number A, binding of primers to the single stranded DNA, number B, chain elongation by DNA polymerase, number C, denaturation of DNA, and number D, none of these. So, which is the first step in the PCR technique? Try to answer the question in the chat box. Even if it is wrong, it does not matter, but I would like to request all of you to try. Otherwise, I won't understand how much of the course you have understood, or which things I need to emphasize much. So, what is the first step in the PCR technique? Let Let me check the chat box. Okay, so I have muted all of you, and also I have you muted the video so that there is a smooth streaming of the class. So if you have to say anything, you just let me let me know in the chat box. Okay, if you have anything to say, you can write in the chat box. So I think I have one answer, which is number C. Now let me check which is the correct answer. So it was number 4. So the correct answer of the problem number 5 is number C. Very good. Denaturation of DNA. So what is the PCR technique and how it happens? So as you can see, this is the step 1 of the PCR. So PCR means polymeric chain reaction, right? So we all know this is a very important technique in many enzymatic applications. So the first step for the PCR technique, it will be denaturation of DNA, and how it will be happening? We have to rise the temperature up to 95 degrees centigrade. So once the temperature is 95 degrees centigrade, so once the temperature is 95 degrees centigrade, what will happen? The double stranded DNA, they will separate. Okay, the double helix, it will be breaking, and when the helix is breaking, we will have two DNA strand. One is from 5 prime to 3 prime, another one is from 3 prime to 5 prime, and this 5 prime to 3 prime strand, it is called the DNA template strand. Okay, so from parent DNA from the two DNA strands are formed. One is DNA template strand, which is 5 prime to 3 prime, and the step is denaturation of DNA, and the temperature is 95 degrees centigrade. So now we will have some DNA primers. Okay, this are the DNA primers. There are short base pair and sequences. Okay, and then we will have the DNA polymerase enzyme. So DNA polymerase enzymes are required in the step 2, which is the annealing step, and the temperature of this step 2 or annealing step is 55 degrees centigrade. Now what will happen in the annealing step? There are nucleotides here. As you can see, there is nucleotides DTP, DCTP, and this is nothing but the D, it is for the, okay. So these are the DNTPs, which I have mentioned earlier also. Okay, so DNPTPs are the dinucleosides triphosphate. Okay, so like this is for thymine, this is for cytosine, this is for adenine, this is for guanine. So these primers or the nucleotides, they will come and they will form this section of DNA can be amplified. Okay, so one section of DNA which is formed here, it can be amplified here, and then there is an enzyme which is TAQ enzyme, and this is synthesis phase. So synthesis phase, the temperature is 72 degrees centigrade, and this enzyme, they joins all the nucleotides to form another chain. Okay, and these are the new DNA strands. So this cycle will be repeated for the 20 to 40 times again, and from the two strand, okay, or from one parent DNA, we are getting 4 DNA strand or 2 new double helix strands. So what are the steps? Again I will repeat. The first step is the denaturation, which happens in 95 degrees centigrade. Step 2 happens, which is aniline, which happens in 55 degrees centigrade, and step 3 is the synthesizing step. The enzyme which is involved here is DNA polymerase, the TAQ, and here we will have the new 2 strands of DNA, and the synthesis phase, the temperature is 72 degrees centigrade. So for PCR, you have to remember what are the steps of the PCR, the step 1, step 2 and step 3, and also you have to remember the temperature of each step. Like the first step, it for the temperature for the first step, it is the 95 degrees centigrade. The temperature for the second step, it is the 72 degrees centigrade, and 55 degrees centigrade for the second step, and for the third step the temperature is 72 degrees centigrade. Okay, so this was for the for about the PCR. So the first step of PCR technique is denaturation of DNA, which is option number C. So now moving forward, problem number 6, it says that which of the following statements is false regarding the PCR primers? Your options are: number A, DNA polymerase requires primers to initiate the replication. Number B, PCR primers are short and single stranded. Number C, primers are sequence specific bind to particular genome sequence, and number D, higher the length of the primer, lesser will be the selectivity. So, which of the following statement is false regarding the PCR primer? Read all the options carefully and then answer. Your options are: number A, DNA polymerase requires primers to initiate replication. Number B, PCR primers are short and single stranded. Number C, primers are sequence specific, bind to particular genome sequence. And number D, higher the length of the primer, lesser will be the selectivity. So, which of the following is the correct answer, or which of the following statements is false regarding the PCR primers? Okay, let me check the chat box if there is any answer for the question. Okay, I have few answers. One is number C, another is number B. Okay, let me check which of the following is correct. So, the correct answer for the problem number 6 is, or the false statement regarding the PCR primers are number D, higher the length of the primers, lesser will be the selectivity. So, this statement is false. So, let us read about the primers. So, consideration about primers. So, specificity, specific for the intended target sequence, okay, and the thoughts for the primer design should be the specificity like uniqueness, length, base composition and internal stability. The stability, form stable duplex with template under the PCR conditions like melting temperature, annealing temperature and internal structure. And compatibility, primers used as a pair shell work under the same PCR condition like primer pair making. So, these are the consideration while you are designing the primer. So, general rules for the primer design: primer length and amplicon length. Okay. Primer length determines the specificity and specifically affect its annealing to the template. So, if it is too short, there is low specificity resulting non-specific amplification. Too long, decrease the template binding efficiency at normal annealing temperature due to the higher probability of forming secondary structures such as hairpins. Optimal primer length: 18 to 24 base pairs for general applications, 30 to 35 base pair for multiplex PCR. Optimal amplicon size: 300 to 1000 base pair for general application, avoid greater than 3 kb, 50 to 150 base pair for real time PCR, avoid greater than 4 base pairs. So, these are few things which you have to consider while designing the PCR primers. So, remember, if the DNA polymerase requires primers to initiate replication, it is correct statement. Okay. So, this is correct. PCR primers are short and single stranded, it is also correct. Primers are sequence specific, bind to particular genome sequence, it is also correct. But this statement like number D, higher the length of primer, lesser will be selectivity, it is false. Rather, if the length of the primer is short, the specificity will be lesser or the selectivity will be lesser. But if the length of the too long, it will also decrease the specificity. So, it should not be too small, too short or too long, okay, it should be in between. And for general applications, it should be about 18 to 24 base pair, okay, and 30 to 35 base pair for multiplex PCR. Okay. So, this was your problem number 6. So, again I am saying, if you have any doubt, any query, you can let me know in the chat box. Okay. I will now move toward the next question. So problem number 7, it says that suppose you are doing a PCR to amplify the portion as indicated in red on the following piece of DNA. How many copies of the amplified portion of double strand DNA will be obtained after 10 cycles? Your options are number A 2 to the power 10, number B 2 to the power 8, number C 2 to the power 6 and number D 2 to the power 9. So, how many copies of amplified portion of double strand DNA will be obtained after 10 cycles? So, I have shown you in the, I think in to last slide, that if I have one DNA single piece of DNA, after so it has two double helix. In double helix there are two single strand DNA, and after the amplification what happened, there are 4. So, use that also, you can use that theory also. So, how many PCR will be there? Sorry. So, is there any answer in the chat box for problem number 7? I think I have few answers. Number A. So, number A is the most given answer. Okay, let us check if number A is correct or not. See, there is no marks on these questions, so it does not matter if your answer is wrong. So, the correct answer will be number B, 2 to the power 8. So, always remember, like if there is n number of cycle, so you will get actually 2 to the power n minus 2. Okay, so this is the formula. So, if there is n number of cycle, okay, if the cycle number is n, so you will get the template, so you will get the amplified portion, the copies, it will be 2 to the power n minus 2. So, say here we have 10 cycles, so we will get 2 to the power 8. Okay, so this is the formula for this PCR amplification portion calculation. So, let us discuss the PCR little bit. Okay, so RT-PCR, it stands for the reverse transcriptase. Okay, I will just erase this part. Okay, and I will just write it here. So, remember, if the cycle number is n, so the formula is 2 to the power n minus 2. Okay, so this is the formula. So, now let us discuss little bit about the PCR. So, RT-PCR stands for reverse transcription polymerase chain reaction. It is a technique which is used in genetic studies that allows the detection and quantification of mRNA. Sensitive method that shows whether or not a specific gene is being expressed in a given sample. RT-PCR is often confused with qPCR by students and scientists alike. However, they are separate and distinct techniques. RT-PCR is used to qualitatively detect the gene expression through the creation of complementary DNA or cDNA transcripts from the RNA. qPCR used to quantitatively measure the amplification of DNA using fluorescent probes. So, this is the difference between RT-PCR and qPCR. It is also very important point. So, remember that RT-PCR used to qualitatively detect gene expression through the creation of complementary DNA or cDNA transcripts from RNA. But qPCR it is used for quantitatively measure the amplification of the DNA using fluorescent probes. So, what is the principle in RT-PCR? The RNA template is first converted into the complementary DNA or cDNA using a reverse transcriptase enzyme. The cDNA is then used as a template for exponential amplification using PCR. Okay. So, one step RT-PCR versus two step RT-PCR. In one step approach, the entire reaction occurs in a single tube. But in two step reaction, the reverse transcriptase reaction and PCR amplification be performed in separate tubes. Okay. So, this is the difference between one-step RT-PCR and two-step RT-PCR. So, what is the difference between RT-PCR and traditional PCR? Although RT-PCR and the traditional PCR both produce multiple copies of particular DNA isolates through the amplification, the applications of the two techniques are fundamentally different. The traditional PCR is used to exponentially amplify target DNA sequences. RT-PCR is used to reverse transcript mRNA to complementary DNA and then amplify this result using traditional PCR. There are several reasons why researchers would use the opposed to PCR. Since mRNA is the message that is sent for translation, RT-PCR can give us a measurement of gene expression that PCR cannot. Thus, complementary DNA produced does not contain the introns that DNA does. Therefore, RT-PCR can provide us with genes that may be inserted into the prokaryotes which cannot and do not splice the introns out. This technique is also used for diagnosing genetic disease as well as studying certain viruses whose genetic information are exclusively composed of RNA. So, these are all about the RT-PCR and PCRs. Okay. So, PCR is a very important technique, and just read these things when I will share this PPT with you. Read these things carefully, and I think you can answer any question related to PCR now. So, now I will move toward the next problem. Problem number 8 says, which of the following statements is false regarding the plasmid PBr322? Your options are: number A, PBr322 plasmid contains 2 genes. Number B, it is natural plasmid. Number C, one gene confers resistance to ampicillin. And number D, other gene confers resistance to tetracycline. So, which of the following statements is false regarding plasmid PBr322? Read the options carefully and write for which of the following you think is correct in the chat box. Even if your answer is incorrect, it does not matter, because there is no marks in your these class questions, right? But if you answer this question, it will help you to understand the course better. Okay. So, again I will repeat the problem number 8. It says that which of the following statements is false regarding plasmid PBr322? Your options are: number A, PBr322 plasmid contains 2 genes. Number B, it is natural plasmid. Number C, one gene confers resistance to ampicillin. And number D, other gene confers resistance to tetracycline. Try to answer the question in the chat box. Even if your answer is wrong, it does not matter. Which of the following statements is false regarding plasmid PBr322? Let me check the chat box now. Okay, so I have few answers. It is number B. So, now let us see what is the correct answer and then let us discuss. So, the correct answer is, yeah, the statement number B, which says it is the natural plasmid, it is false, because plasmid PBr322, it is not a natural plasmid. So, PBr322 is a purpose built plasmid vector and was one of the first widely used cloning vector. Let me highlight the things, it will be easier for you. So, it is a purpose built plasmid vector. Okay, it is a purpose built plasmid vector and one of the first widely used cloning vector. It has relatively small size of 4363 base pair. This is important because transformation efficiency is inversely proportional to size and above 10 kbp is very low. Okay. So, this has a very relatively small size and the small size is important because the transformation efficiency is inversely proportional to the size and above 10 kbp it is very low. So, nomenclature of PBr322. Sorry. So, what is the reason for the nomenclature of this particularly PBr322? So, it is created in 1977 in the laboratory of Herbert Boyer in the University of California in San Francisco. So, P it indicates as plasmid. So, P stand for plasmid, BR it identifies the Bolivar and Rodriguez, the two researcher who has developed it. Okay. 322 it distinguishes those plasmids from others, like there are PBr 325, 327, these are developed in the same laboratory. So, this 322 is particularly basically a distinguishing number which distinguishes this particular plasmid from the another plasmid. So, PBr 322 is a 4361 base pair in length and has 2 antibiotic resistant genes. It is very important. Okay. So, to remember, like it has 4361 base pair and it has base pair in length and has 2 antibiotic resistant gene. The gene BLA encoding the ampicillin resistance protein and the gene TETA encoding the tetracycline resistant protein. Okay. So, there are 2 genes, one is BLA, they encodes for the ampicillin resistant protein and gene TETA it encodes for the tetracycline resistance protein. It contains the origin of replication PMB1 and the ROP gene which encodes the restrictor of the plasmid copy number. Okay. The plasmid has a unique restriction sites for more than 40 restriction enzyme. So, plasmid has unique restriction site for more than 40 restriction enzymes. 11 of these 40 sites lies within the TET gene. There are 2 sites for the restriction enzymes HIND3 and CLA1 within the promoter of TRT gene. There are the 6 key restriction sites are amplifier gene. So, in short, as you can see here in the picture, it is 4.4 kb this base pair, there are 4361 base pair, so which is 4.4 roughly, and there are E. coli and hind I here in the start. Then we have TETR gene which encodes for the tetracycline resistant protein, and then we have this AMPR which encodes for the particularly ampicillin resistant protein. So, it is isolated from E. coli, remember it is isolated from E. coli. The size 4361 base pair, cloning limit 0.1 to 10 kb, marker gene ampicillin and tetracycline resistant gene, restriction site for various restriction endonucleases. Okay. So, remember this few things about PBr322. So, PBr322 is a very important plasmid. Okay. So, I would request all of you to read these things very carefully and remember these things, okay, because it is a very important topic for your molecular biology portion. Okay. So, now I will move toward the problem number 9. So, problem number 9 says consider the following diagram and identify the correct statement. Okay, as you can see there is a diagram, one is 3 prime to 5 prime which is strand A and 5 prime to 3 prime it is strand B. So, there is a D1, D2 and there is a strand C also and X also. So, from this diagram you have to answer that number A, B is the template strand, number B, A is the primer, number C, C is the non-coding strand and number D, A is the sense strand. So, which of the following is correct? Identify from the diagram. So, write in the chat box which of the following you think is the correct. Considering the following diagram, B is the template strand, A is the primer, C is the non-coding strand and A is the sense strand. So, which of the following is correct? Let me know in the chat box. It does not matter even if your answer is incorrect, but at least all of you should try to answer this. I have discussed it in the last class also, as I remember. So, I hope many of you will answer this question. Considering the following diagram, identify the correct statement. B is the template strand, A is the primer, C is the non-coding strand, A is the sense strand. Which of the following is correct answer? Let me see the chat box. Okay, so I have answers like number D, number A, number A. So, I have either D and or A. So, let me check which of the following is correct. So, the correct answer will be number D. A is the sense strand. Okay, it is correct. So, as you can see, the strand which runs from 3 prime to 5 prime, it is the anti-sense strand. Okay, and the strand which goes from 5 prime to 3 prime, it is the sense strand. Okay. Okay, I think there is a mistake. Let me check once. Okay, I think there is a typographical error here. Okay, I think there is a typographical error here. Okay, so I will just correct it later. Okay, so I think there is a error here because strand A is not the sense strand. The sense A is the anti-sense strand. Okay, so there is a error here. Okay. In fact, this is, let me just correct it. So, one thing, there is a error here. So, your strand A, it will be your anti-sense strand. Strand A. So, the strand A, it runs from 3 prime to 5 prime, and the strand which goes for 3 prime, okay, there is a mistake in this picture also, I think, because here it is saying that the sense strand will be from 5 prime to 3 prime, but here it says that the sense strands will direct it from 3 prime to 5 prime. So, I think there is a mistake in this picture or this picture, this table also. So, I will check and I will just let you know. I think this is also because A strand is the sense strand, it should be. Okay. So, okay, anyways, I will just correct it before uploading. So, do not worry, just remember few things about the sense strand and anti-sense strand. So, sense strand, it is directed from 3 prime to 5 prime direction. Okay, and anti-sense strand, it directed from the 5 prime to 3 prime. So, it is no sense strand, it does not transcripts into mRNA, but anti-sense strand it is transcribed into mRNA. The sense strand contains same nucleotide sequence as mRNA except thymine, and it contains the complementary nucleotide sequence than mRNA. It contains codons, it contains anticodons. There are no hydrogen bonds are formed between the sense strands and synthesizing mRNA, and the nucleotides are in the antisense strands are temporarily hydrogen bonded with the complementary nucleotides in the synthesizing mRNA. Okay, so I think there is a mistake, there is a confusion here, but I think this the previous answer was correct. A was the sense strand. Okay, so anyways, I will just check it later also once I finish the class, and I will just let you know. Okay, I will just update this PPT before uploading, so don't worry. So according to this figure, the strand goes from 3 prime to 5 prime, it is the sense strand. Okay, and as you can see, the strand A, it goes from 3 prime to 5 prime, so it is the sense strand, and it is the antisense strands. Also I think strand B, it can be the template strand. So option number A can also be correct. So this problem number 9, it has some problem, I will just correct it before uploading. Okay, so moving toward the problem number 10, it says that which of the following statement is false regarding cystic fibrosis? Your options are: number A, it is a genetic disease; number B, PCR can detect the disease; number C, in non-cystic fibrosis individual or CF individual, CTFR gene codes for a protein that is sodium ion channel. Okay, and number D, in CF individual, mutation in CTFR gene leads to thick mucus secretion in lungs. So which of the following statements is false regarding the cystic fibrosis? It is also a part of your molecular biology, the cystic fibrosis disease. So your options are: number A, it is a genetic disease; number B, PCR can detect the disease; number C, in non-CF individual, CTFR gene codes for a protein that is sodium sodium ion channel, and in CF individual, mutation in CTFR gene leads to thick mucus secretion in lungs. Sorry. So which of the following statements is false regarding cystic fibrosis? Is there any answer? Problem number 9, I will correct it before uploading, so don't worry, there is some problem there. So the correct options, I have a doubt here, I will just let you know later. So the correct answer for problem number 10 is: in non-CF individual, CTFR gene codes for a protein that is in sodium ion channel. Okay, so this is the false question or false option. So let us discuss the gene mutation in cystic fibrosis. Okay, so cilia move back and forth here in the lungs, so chloride ions. So for normal DNA, okay, the CFTR protein develops normally, reaches the cell surface and becomes an open channel or door for the chloride ions. Okay, so the door jamming mutation including G551D. So as you can see, there are chlorides ion here. This is the cell wall, this is the cilia, this is the CFTR protein. Okay, and this protein, it codes for not sodium ion channel but for the chloride ion channel. So that's why your option number 3, it is incorrect, because it is not the sodium, it is for the chloride ion channel. Okay. So then chloride ions, so if there is a this door jamming for mutation including G551D, the mutation affecting LoRa and Ket cleavage disable function at the cell surface. The chloride ion can reach the channel at the cell surface but cannot pass through it. So this has been caused a Doorman problem. So, there will be a thick sticky mucus buildup that flattens the cilia. Okay. The mutant CF proteins needs a corrector drug to boost it to the surface. It also needs a dormant drug to open the channel so that the chloride ions can pass through. Okay, so CF lung cell within two problems of location and function. So, common delta 5 mutation. So, it is this mutation which is CTFR. It leads to the thick secretion. Okay, and this thick secretion, it will actually will form a mucus and it will be flattening the cilia, and that is how it works. So, remember the problem number C is incorrect because the non-CF individual, the CTFR gene encodes for the chloride ion channel. Okay. So, now I will move toward the next problem. So, problem number 11, it says that which of the following statement is false regarding eukaryotic mRNA? Your options are: number A, they are derived from larger RNA precursors; number B, they result from extensive processing of the primary transcripts before serving as translation components; number C, they usually have poly A tails at their 3' end, and they have both introns and exons in the mature form. So, which of the following statement is false regarding the eukaryotic mRNA? They are derived from larger RNA precursors. Option B says they result from extensive processing of their primary transcripts before serving as translation components. Number C, they usually have poly A tails at their 3' end. And number D, they have both introns and exons in matured form. Now, let me check the chat box. All of you should try to answer these questions. Which of the following statement is false regarding eukaryotic mRNA? They are derived from larger RNA precursors. They result from extensive processing of their primary transcripts before serving as translation components. Option C, they usually have poly A tails at their 3' end. And option D, they have both introns and exons in matured form. So, which of the following statement is false regarding eukaryotic mRNA? Is there any answer in the chat box? Let me check. I have one answer which says it is number A. So, let us check. So, the correct answer will be number D. So, number D statement, it is false. Okay. Sorry. So, RNA polymerase II transcripts long precursor RNAs. The pre-mRNA must be spliced or processed into mature mRNA. So, in mature mRNA, so first in the pre-RNA, so they are derived, this eukaryotic mRNA, they are derived from large precursor RNAs, okay, and they result from the extensive processing of the primary transcripts. And in the initial your precursor RNA, so it has the exons and introns, I have discussed like in the last class also, like the exons are important, but the introns are not necessary, and there is an enzyme which is called the spliceosome, which remove the introns and joins the exons to make the spliced mRNA or mature mRNA for the pre-mRNA. Okay. So, option number D, which says that they have both introns and exons in mature form, it is false regarding the eukaryotic mRNA. So, now what are the differences between the prokaryotic mRNA and eukaryotic mRNA? In mRNA produced by transcription for operons in prokaryotes, and the mRNA produced by the transcription in single genes in eukaryotes. The synthesis of mRNA occurs in the cytoplasm. The synthesis of mRNA occurs inside the nucleus for the eukaryotic mRNA. The prokaryotic mRNA, it is polycistronic, okay, and the eukaryotic one is the monocistronic. So, the prokaryotic mRNA does not contain both 5 prime cap and 3 prime tail. The eukaryotic mRNA contains both 5 prime cap and 3 prime tail. The transcription is coupled with translation in prokaryotes. The transcription of the eukaryotes occur after the completion of the transcription. The prokaryotic mRNA, it contains the ribosome binding site, which is Shin-Dalgarno sequence, okay, and the 5 prime cap in the eukaryotic mRNA is responsible for recruiting ribosomes. Lifespan of the prokaryotic mRNA is very short. Lifespan of the eukaryotic mRNA is relatively stable. The prokaryotic mRNA undergoes a very little post-translational modification, but the eukaryotic mRNA undergoes major post-transcriptional modifications. So, these were the comparisons for the prokaryotic mRNA and eukaryotic mRNA. So, remember in the first there is a precursor RNA, and RNA polymerase II, it transcripts long precursor RNA into pre-mRNA, and then spliceosome, they remove the introns and join the exons to make a mature mRNA. So, moving toward our next problem. Problem number 12 says, in Huntington disease, the trinucleotide sequence that repeats more than 36 times is number A CAA, number B AGC, number C CAG, and number D TAC. So, which of the following trinucleotide sequence repeats more than 36 times in Huntington disease? Try to answer this question in the chat box. So, which of the following options is correct? In Huntington disease, the trinucleotide sequence that repeats more than 36 times is number A CAA, number B AGC, number C CAG, and number D TAC. Which of the following is correct? Try to answer it in the chat box. Even if your answer is wrong, it does not matter, but all of you should try to answer it. Let me check the chat box if there is any answers. Okay, I have one answer which says problem number D. Okay, let me check. So the correct answer will be number C, CAG. This trinucleotide sequence repeats more than 36 times in Huntington disease. So as you can see, there is normal blood sample. If you do the PCR and if you find CAG, it is repeating more than 36 times, then then we have to do the Sanger sequencing. Okay, and in the Huntington disease, the extended CAG sequence is there. Okay. What are the clinical features for the Huntington's disease? Like for motor, there is a chorea, dystonia, motor impersistence, dysrhythmia, dysphagia, poor posture, incoordination, impaired gut and balance, delayed initiation of voluntary success. Okay. For cognitive, loss of executive functions, difficulty in multitasking, rigid and inflexible thoughts, poor concentration, easily distracted, impaired spatial perception, impulse control and disinhibition, depreciation of vocabulary, poor insight, short term memory loss. Psychiatric, there is depression, anhedonia, decreased libido, suicidal ideation, social isolation, disrupted sleep pattern, delusion, hallucination, paranoia, and irritability. So these are the clinical features for the Huntington disease. So you don't need to memorize everything, just remember few clinical features and remember this CAG, this trinucleotide sequence repeats more than 36 times for the Huntington disease. Okay. Now moving toward the next problem. Problem number 13 says identify the incorrect statement among the following. Your options are: number A, cofactors are either small organic molecules or inorganic metal ions. Option B, an enzyme without cofactor is called hollow enzyme. Option C, prosthetic groups are coenzymes which binds permanently with the enzymes. And number D, cofactor is non-protein molecule. So which of the following is the incorrect statement regarding cofactors and enzymes and coenzymes? Option number A, cofactors are either small organic molecules or inorganic metal ions. Option B, an enzyme without cofactor is called hollow enzyme. Option C, prosthetic groups are coenzymes which binds permanently with enzymes. And option D, cofactor is non-protein molecule. Which of the following statement is incorrect? Read the options carefully. It is very easy. I know you can do it. So all of you try to answer this question in the chat box. Even if your answer is wrong, I want all of you to try to answer this question, because it is a very basic question regarding your cofactors, enzyme, coenzymes and everything. Everybody try to answer this question. Identify the incorrect statement among the following. Cofactors are either small organic molecules or inorganic metal ions. An enzyme without cofactor is called hollow enzyme. Prosthetic groups are coenzymes which binds permanently with enzymes. And cofactor is non-protein molecule. Which of the following statement is incorrect? Let me check the chat box. Okay, so I have two answers. One it is saying it is number B and one is number D. Okay, let me check which of the following is correct. Okay. So, the correct answer or the incorrect statement among the following will be number B, an enzyme without cofactor is called hollow enzyme. So, this statement is false. So, why this statement is false? Now, we have to discuss. So, coenzymes and cofactors, these are very important terms for your enzyme studies. Okay. A large number of enzymes require an additional non-protein component to carry out its catalytic functions are called cofactors. A coenzyme or metal ion that is covalently bound with the enzyme protein is called prosthetic group. So, cofactors are two types. One, there is inorganic ions such as iron, magnesium, manganese, zinc. A complex organic molecule is called coenzyme. So, cofactor. So, coenzyme is basically one type of cofactors, and cofactors can be inorganic ions or organic molecules. So, your option for first option which says that cofactors are either small organic molecules or inorganic metal ion, it is correct. An enzyme without cofactor is called hollow enzyme. So, now it is the statement where we have to focus. So, some enzyme require both coenzyme and one or two more metal ions for their activity. Coenzymes function as transient carriers of specific functional groups. Okay. So, there is an enzyme which is very a protein portion or inactive, which is called a apoenzyme, and when cofactor bounds or coenzyme or cofactor which is a non-protein portion, it acts as a activator, and there is a your hollow enzyme or a whole enzyme or a active enzyme, okay, which is a complete enzyme. So, an enzyme without cofactor is not called hollow enzyme, it calls what? It calls apoenzyme. Okay. So, this statement is incorrect. Either you can say that the enzyme with cofactor is called hollow enzyme, it will be correct, or an enzyme without cofactor is called apoenzyme, okay, which is a inactive form, which is only protein portion, and when cofactors binds with the apoenzyme, it forms the hollow enzyme, or which is the complete whole enzyme which is active. So, differences between the cofactor and coenzymes is also very important. So, let me erase this. Okay. So, what are the differences between the coenzyme and cofactor? Coenzyme is a small organic non-protein molecules that carry chemical groups within between the enzymes, and cofactor is a non-protein chemical compound that is tightly bound and loosely binds with an enzyme or other protein molecules. So, coenzyme is a type of cofactor. Coenzymes and prosthetic groups are two types of cofactors. So, the coenzymes are basically molecules and cofactors are chemical compound. Coenzymes are organic molecule, cofactors mostly are inorganic compounds. So, these are loosely bound with enzyme which are coenzyme, some are covalently bound to the enzyme which are cofactor. They assist biological transformation, they aid the function of the relative enzyme, they serve as a carriers to the enzymes, they increase the rate of the reaction that is catalyzed by the enzyme. These can be removed from the enzyme very easily. These can only be removed by the denaturing the enzyme. For example, there are vitamin, biotin, coenzyme A, these are the coenzymes, and examples of cofactors include metal ions like zinc 2+, potassium 2+, magnesium 2+. Okay. Cofactor also can be complex organic molecules, but mostly these are called the coenzymes. Okay. So, you can remember cofactors of two types, one is inorganic ions and another is your organic molecules, big organic molecules which are called the coenzymes. Okay. So, these are, and remember the inactive protein portion of a native enzyme is called apoenzyme, and when cofactor bounds which is a non-protein portion, it will activate the apoenzyme and it will form a complete whole enzyme which is called the hollow enzyme. Okay. So, this was the problem number 13. I will now move toward the next problem. Problem number 14 says identify the correct statement among the following. Your options are number A, vitamin B is lipid soluble vitamin. Option B, water soluble vitamins act as a enzyme cofactors. Option C, vitamin E act as a precursor of coenzymes. And option D, regular intake of vitamin C is not necessary for our body. So, which of the following is correct statement among the following options? Option A, vitamin B is lipid soluble vitamin. Option B, water soluble vitamins act as enzyme cofactors. Option C, vitamin E act as a precursor of coenzymes. And option D, regular intake of vitamin C is not necessary for our body. Identify the correct statement among the following. All of you try to answer this question. So, identify the correct statement. Which of the following is? Number A, vitamin B is lipid soluble vitamin. Number B, water soluble vitamins acts as coenzyme cofactors. Let me check. I think somebody wants to join. So, I have any answers? I have answer number option number B, D and option number A. So, I have 3 options here as answers. Okay. So, let me check which of the following is the correct answer, or any one of you, if others can want to try, you can try. Identify the correct statement. Option A, vitamin B is lipid soluble vitamin. Option B, water soluble vitamins act as a enzyme cofactors. Option C, vitamin E act as a precursor of coenzymes. And option D, regular intake of vitamin C is not necessary for our body. So, the correct answer will be, somebody wants to join again. So, I have few answers like number B, D, A and D. So, let me check now which of the following is the correct answer. So, the correct answer will be number B, water soluble vitamins act as a enzyme cofactors. Option B is not a lipid soluble vitamin. Option vitamin B is a actually water soluble vitamin. There are lipid soluble vitamin, always remember there are 4 vitamins which are lipid soluble, which are A, D, E and K. ADEC, you can remember it like it also. So, vitamin A, D, E and K, these are lipid soluble, okay, and all other vitamins are your water soluble vitamins. So, that is why problem option number is incorrect. Water soluble vitamins can act as enzyme cofactors, it is correct. Vitamin E act as a precursor of coenzyme, it is not correct, okay, because it is a lipid soluble vitamin and it cannot act as a precursor of coenzyme. Regular intake of vitamin C, it is not necessary for the body, it is not correct statement. We have to have vitamin C, otherwise we will have various diseases like scurvy. It is a very well known disease for the gum or bleeding teeth, okay. Vitamin C is also very necessary for our body's immunity. So, option number D is also incorrect. So, now let us discuss about coenzymes and vitamins. So, most of the B vitamins are coenzymes and are essential in facilitating the transfer of atoms or groups of atoms between molecules in the formation of carbohydrate, fats and proteins, okay. Coenzymes, they are complex organic molecules that work with the enzymes to facilitate the enzyme's activity. Many coenzymes have B vitamins as a part of their structure. Coenzymes is to act as transporters of chemical groups from one reactant to another. Examples include nicotinamide adenine dinucleotide or NAD, which accepts the hydrogen and gives it up into the another reaction. ATP or adenosyl triphosphate, which gives up phosphate groups while transferring the chemical energy. So, vitamins, coenzymes and cofactors, primarily they are organic molecules. They may have inorganic component or cofactor. Some work with or are part of an enzyme. They promote or catalyze the biochemical reactions. Most cannot be made by human body. Water soluble or all vitamin B and vitamin C. Vitamin A, D, E, K are lipid soluble. Most of the vitamin B are used in secondary metabolism. And what is the purpose of these lipid soluble vitamins? For vitamin A, the purpose is vision, growth, development and immune function. The deficiency of vitamin A will lead to night blindness, heart and scaly skin, reproductive issues and increased infection. Toxicity, if you have too much vitamin A, it is not also very good, because in food there is rare supplements, headache, scaly skin, liver damage and birth defect. The sources: food is the liver, yellow and orange vegetable, green leafy vegetable. Vitamin D for bone growth, development in children, bone maintenance in adults, bone calcium regulation, insulin formation and secretion. The deficiency will lead to rickets, osteomalacia or soft bone, osteoporosis or porous bones. The toxicity will lead to hypercalcemia or high blood calcium. The synthesis: sunlight, skin exposure or vitamin D fortified milk and cereals. Vitamin E, it is an antioxidant and it is enhanced by the vitamin C and selenium. Rare from diet, it can deficiency fat malabsorption syndrome, and preterm infant can have this vitamin E deficiency. The toxicity is rare. Okay, large supplement doses can increase the risk of bleeding in person with vitamin K deficiency. And the fruit, animal products, wheat germ oil, vegetable and seed oils. Vitamin K, its purpose is blood clotting and bone formation. The deficiency rare in adults, fat malabsorption syndrome, prolonged antibiotic use, okay, mega dose of vitamin A and vitamin E. The toxicity, the rare from food, hemolytic anemia in newborns, food, plant foods, green leafy vegetables, soybean, canola, olive oil synthesis, these are the sources. So, now moving toward the next problem. Problem number 15 says oxidation of retinol to retinal is catalyzed by which enzyme? Your options are: number A, retinal dehydrogenase, number B, retinal reductase, number C, retinal isomerase and number D, retinal deoxygenase. So, which of the following is correct? I want all of you to try to answer this question in the chat box. Oxidation of retinol to retinal is catalyzed by which enzyme? Retinal dehydrogenase, retinal reductase, retinal isomerase or retinal deoxygenase. So, which of the following is the correct answer? Oxidation of retinol to retinal is catalyzed by which enzyme? Retinal dehydrogenase, retinal reductase, retinal isomerase or retinal deoxygenase. Is there any answer in the chat box? Okay, so I think I have few answers. It says number A, number D, number A. So I have most the number A's and one number D. Okay, let me check. See, answering the questions is very important, even if your answer is wrong, but still you guys are answering, which is very important. So the correct option is number A. Very good. It is retinal dehydrogenase. Okay. So now let us discuss it. So all this is your vitamin A reserves. So, okay. So this is the all trans retinal ester. Okay. So this is called all trans retinal ester. So there is a ester hydrolase enzyme which converts this all trans retinal ester to all trans retinol. Okay, this is the all trans retinol. So this all trans retinol is converted to all trans retinal by the enzyme retinol dehydrogenase. Okay, or the by the enzyme retinal or retinol dehydrogenase, you can say both are correct. Okay, so there is a Vision cycle here. Okay, so if there is a 11 cis retinal Vision, so Vision cycle reaction happens like this. So if this is a 11 cis, this indicates the Vision. Okay, and this 11 cis retinal, it converted to all trans retinal by the H nu. Okay, and there is a alcohol dehydrogenase which convert this all trans retinal to the all trans retinoic acid. So this remembering these things are very easy in one way. So if there is a retinol, okay, retinol means it is your alcohol, retinal means it is aldehyde, and retinoic acid means there will be acid, and this 11 cis retinal it is important for the vision. So remember this thing. So option A, it is correct. Okay, retinal dehydrogenase is the enzyme which actually converts this retinol to retinal. So now I will move to the next problem. Problem number 16 says which of the following is the coenzyme form of vitamin plantothanic acid? Number A, pyridoxal phosphate, number B, tetrahydrofolate, number C, thiamine phosphate, and number D, coenzyme A. So which of the the following is the coenzyme form of the plantothanic acid, vitamin plantothanic acid? So this plantothanic acid is one very important vitamin. So there is a short form also, but remembering the full form of the vitamins or the larger form of the vitamin is also important. As we all know, the ascorbic acid means vitamin C. So like this, plantothanic acid is one of the some vitamin. So I will discuss it later. So which of the following is the coenzyme form of vitamin plantothanic acid? One, option A is pyridoxal phosphate, option B tetrahydrofolate, option C thiamine phosphate, and option D coenzyme A. So which of the following is the coenzyme form of vitamin plantothanic acid? Try to answer this in the chat box. Even if your answer is wrong, it does not matter, but all of you should try to answer these questions. Anyways, I will discuss it in detail, but if you answer, it will help you to understand the course better. So which of the following is the coenzyme form of vitamin plantothanic acid? And also, if possible, or if you know, you can write what is the which vitamin plantothanic acid means which vitamin. Also try to write it in the chat box. Like ascorbic acid is the vitamin C, all of you know. So plantothanic acid is which vitamin? Write it in the chat box along with the right option which you think is correct for the problem number 16. Plantothanic acid means which vitamin and what is the correct option of this problem number 16. Let me check the chat box now. Vitamin B3, I have one answer, vitamin which says it is vitamin B3, and your another options are I think number D, number C, number D. Okay, now let me check which is the correct answer. So the correct answer is number D, coenzyme A. So vitamin B3 actually the full form is niacin. Vitamin B5 is the plantothanic acid. So let us discuss what is the vitamin, what is the coenzyme, and what is the group which is transferred here. Okay, so vitamin B1. Okay, so this is the for the your B complex, vitamin B complexes. We all know like vitamin B is, it comes from a complex form. Okay, so there are many vitamins which are togetherly called vitamin B or complex. Like for B1 the full form is thiamine. Okay, and the coenzyme is thiamine pyrophosphate or TPP. The group transfer or the group which is transferred by it is C2-aldehyde, decarboxylation and decarboxylation happens here. Okay, B2 is the riboflavin. The coenzyme is flavin adenine mononucleotide or FMN, and the group which is transferred is hydrogen. Or flavin adenine dinucleotide or FAD also is the coenzyme for vitamin B2, and it is the group transfer is also hydrogen. Vitamin B3, it is the niacin, okay, which is nicotinamide adenine dinucleotide or NAD plus or nicotinamide adenine dinucleotide phosphate, which is NADP. The group which is transferred by it is hydrogen. Okay. Vitamin B5, it is the plantothanic acid. Okay, the plantothanic acid is vitamin B5. It is coenzyme A. The coenzyme form for the vitamin plantothanic acid is coenzyme A, and the group which it transferred is the acyl group. Vitamin B6 is the pyridoxine. Okay, the coenzyme is pyridoxal phosphate, and the group which is transferred is amino group, and decarboxylation happens. Vitamin B7 is the biotin. The coenzyme is biocytin, and the group which is transferred is carboxyl. Vitamin B9 is the folic acid. The coenzyme is tetrahydrofolic acid, and the group which is transferred is the formyl group. Vitamin B12 is the cyanocobalamin, which is have the vitamin 12 coenzyme, and the group which is transferred is carboxyl and HX rearrangement, where X is the halogen. So these are the coenzyme forms of various vitamin B's. So remember this chart. Okay, if you don't remember the group transfer is alright, but remember which is the coenzyme for which vitamin. Like for vitamin B1 it is TPP, vitamin B2 it is FMN and FAD, vitamin B3 it is NAD plus and NADP plus, vitamin B5 or plantothanic acid it is coenzyme A, vitamin B6 it is pyridoxal phosphate, vitamin B7 it is biocytin, vitamin B9 it is tetrahydrofolic acid, and vitamin B12 it is cyanocobalamin. Also just remember the names, like vitamin B1 is thiamine, vitamin B2 is riboflavin, vitamin B3 is niacin, vitamin B5 is pantothenic acid, vitamin B6 is pyridoxine, vitamin B7 is biotin, vitamin B9 is folic acid, and vitamin B12 is cyanocobalamin. Okay. So moving toward the problem number 17. Okay, one more thing here, it is your the complete reaction cycle is also here. There is plantothanic acid, it converts to 4' phosphopantothenic acid by plantothanic acid kinase. Okay, and then we have 4 phosphopantothenic cysteine, and here the enzyme is 4' phosphopantothenic cysteine synthesis. It converts to the 4 phosphopantothenic by the removal of carbon dioxide, then to 4' dephosphocoenzyme A, and then coenzyme A. So this is the reaction cycle from plantothenic acid to coenzyme A. Okay, and this is coenzyme A, this is so apoenzyme, and there is a holoenzyme where phosphoplantothenic is the active group. So you don't need to memorize this thing. Okay, you don't need to memorize this whole chart, just for your knowledge I just mentioned it here, like how plantothenic acid is converted to the coenzyme A. Okay, and remember one more thing, this is also very important, like coenzyme A, the apoenzyme, okay, coenzyme A, the apoacyl carrier protein, it is converted to the hollow acyl carrier protein or hollow enzyme with the phosphoplantothaniin as a active group. So, the cofactor is phosphoplantothaniin, which converts this apo coenzyme to hollow coenzyme A. Okay, so this is a very important. So, remember just this thing and remember this full forms and also the coenzymes. So, now next problem, it says that which of the two reactions are required to biosynthesize the first derivative of vitamin D inside the body. The options are number A, disrotatory ring opening followed by 1,5 hydrogen shift, number B, conrotatory ring closing followed by 1,5 hydrogen shift, number C, disrotatory ring closing followed by 1,7 hydrogen shift, and number D, conrotatory ring opening followed by 1,7 hydrogen shift. So, which of the following two reactions are required to biosynthesize the first derivative of vitamin D inside the body? Everybody try to answer this question in the chat box. Which of the following options you think is correct? Option A, B, C or D? Which are the two reactions are required for biosynthesize the first derivative of vitamin D inside the body? Number A, disrotatory ring opening by 1,5 hydrogen shift. Number B, conrotatory ring closing followed by 1,5 hydrogen shift. Number C, disrotatory ring closing followed by 1,7 hydrogen shift. Or number D, conrotatory ring opening followed by 1,7 hydrogen shift. Which two reactions are required for biosynthesize the first derivative of vitamin D inside the body? This question is very important for this course also, or for this coenzyme purpose. This question is very important for your organic chemistry point of view and from also your medicinal chemistry point of view. Which of the following is the correct answer? I want everybody to try to answer this question. It is a very important question. Let me check the chat box. Okay. So, I have two answers, one is number A and another is number D. Okay, let me check which is the correct answer. So, the correct answer for this question is number D. Very good. Conrotatory ring opening followed by 1,7 hydrogen shift. So, look at this answers very carefully because this is a very important question. So, before starting, let us discuss what are the conrotatory ring opening and what is the hydrogen shifts. Okay. So, basically 6-pi electrocyclic ring closing. Okay, let me show the whole slide first. So, as you can see, this is the 7-dehydrocholesterol. Okay. After 6-pi conrotatory ring opening, it forms the pre-vitamin D3, and then after 1,7 hydrogen shift, it forms the vitamin D3, which is the precursor for the vitamin D biosynthesis. Okay. So, as you can see, if you look at here carefully, this ring opening, the first step is a ring opening. Here it is a ring, a 6-member ring, and when it is opened, it is formed like this. Okay. And this ring opening, it has 6-pi conrotatory ring opening because here we have 6-pi electrons. After the ring opening, you can see there are 3 double bonds. One double bond is here, one double bond is here and one double bond is here. Okay. So, if you can look at this double bonds carefully, okay, let me erase and highlight this part. So, it will be easier. So, because there are 6 pi electrons are involved, it is called the 6 pi ring opening. Okay, because after ring opening we are having 6 pi electrons. So, there are few conditions, these are all called the pericyclic reaction, which are the ring opening and ring closing basically. So, there is a peripheral movement of electrons by which the rings opens and ring close. So, the movement of electrons are like this goes there, this comes here and this comes here. So, if the electron flow is like this, we may have this group or we may have this function. Okay. So, there are two conditions for the photochemical reaction, one is thermal condition and one is photochemical condition. So, remember, if the condition is thermal, so the 6 pi electrocyclic ring closure, it will be disrotatory in function, and if the condition is photochemical, it will be the ring closing and ring opening, it will be conrotatory in motion. What is conrotatory, disrotatory? It is not the you can read it from any pericyclic book. If you have any doubt understanding this, you can just email me, I will suggest you a very good books or very where you can read this particular example. Okay. So, since this is all of this happens under the UV condition or sunlight condition. So, we that is why it is said that for vitamin D it is good for having sunlight on your body. Why? Because this particular reaction happens for vitamin D formation, and this is catalyzed by the sunlight. So, under photochemical condition of HNU or sunlight, the 6 pi ring opening and ring closing, it is conrotatory. So, the first reaction is the conrotatory ring opening. And now the second one is followed the 1,7 hydrogen shift. Now, why it is called the 1,7 hydrogen shift? So, this is one. So, if the numbering happens like this is a sigmatropic reaction. So, the numbering happening like this, the bond which is breaking, as you can see here, this bond is breaking. So, this will be 1 and this will be 1, this will be 2, this will be 3, 4 and 5. So, as you can see, the hydrogen is transferred from one position to 5th position, and there now, so it is the 1,5 hydrogen shift. And if this happens like 1 to 7, so it is 1, it is 1, it is 2, it is 3, it is 4, it is 5, it is 6 and it is 7. So, hydrogen is transferring from 1 to 7 position in this ring. So, it is called the 1,7 hydrogen shift. Now, if you look at carefully, this is, there is a hydrogen, okay, and it is CH2 if you write it. So, it is 1, it is 1, it is 2, 3, 4, 5, 6 and 7. The hydrogen is transferred from here to here, okay, from here to here the hydrogen is transferring. So, it is the 1,7 hydrogen shift. So, it is a very important example and very important question also. Okay. So, remember, the first step will be conrotatory ring opening and the second step will be 1,7 hydrogen shift. If you have any doubt understanding this problem, you can write me through email. I will suggest you some good notes and suggest you some good books where you can read it this conrotatory, disrotatory things in detail, because it is not in scope of our course, but this example is very important. Okay. So, now I will move toward the next problem. Problem number 18 says that the conversion of the pyridoxine to PLP is done by the enzymes number A, phosphatase and kinase, number B, kinase and dehydrogenase, number C, oxidase and deoxygenase, and number D, dehydrogenase and kinase. So, the conversion of pyridoxine to PLP is done by which enzymes? Which set of enzymes catalyze the conversion of pyridoxine to PLP? Phosphatase kinase, kinase dehydrogenase, oxidase deoxygenase and dehydrogenase kinase. Identify the correct set of enzymes. So, conversion of pyridoxine to PLP is done by which set of enzymes? There any answer in chat box? Let me check. Okay, I have two answers, number B and number A. Okay, let me check which is the which of the following is the correct answer. So, the correct answer is number D, dehydrogenase and kinase. So, what is PLP? PLP is nothing but pyridoxal phosphate. So, PLP is derived from vitamin B6 family of vitamins. PLP is a coenzyme for enzymes catalyzing reactions involving amino acid metabolism like isomerizations, decarboxylation and transaminations. Okay. So, this is the structure of your pyridoxine. This is the structure of the pyridoxal and it is for the pyridoxamine. So, as you can see here, it is a alcoholic group and it is in pyridoxal, it is the aldehyde group, and in the pyridoxamine it is the amine group here. Okay. Otherwise the structure is same, and the pyridoxal 5 prime phosphate is the pyridoxal phosphate is the PLP, where the 5 prime alcoholic group is phosphatized. Okay. So, these are structures which are related for this vitamin B6 family and PLP. So, first vitamin B6 is pyridoxine is intimately involved in the porphyrin, glycogen, lipid and amino acid metabolism. In plasma, it is found largely as the pyridoxine or pyridoxaldehyde and intercellularly as phosphorylate derivative. So, pyridoxine, it is converted to pyridoxaldehyde, okay, by the NAD plus and NADH, and the enzyme which is involved here is the dehydrogenase. Okay, why dehydrogenase? As NAD plus is converting to NH plus and alcohol is converting to aldehyde. So, basically it is a oxidation, and whenever this kind of oxidation reaction happens, dehydrogenase is the enzyme. Second step is the pyridoxaldehyde to pyridoxal phosphate. Okay, and then since one phosphate group is transferred, so ATP will be converted to ADP. And so, this enzyme which is involved is pyridoxal kinase. So, second enzyme is kinase, which actually transferred one phosphate to the 5 prime of this pyridoxaldehyde to form the pyridoxal phosphate. Okay. And the enzyme kinase, it helps, basically it transfers the phosphate group or gamma phosphate group from ATP to ADP, and then there is a pyridoxamine phosphate, it forms, okay, an amino acid. So, this is the group transfer reaction. Okay. So, the enzyme, correct set of enzymes will be dehydrogenase and kinase, which catalyze the conversion from pyridoxine to PLP. Okay. So, quickly I will move toward the next question. Problem number 17 says, which heterocyclic rings are present in TPP or thiamine pyrophosphate? Your options are number A, pyrazole and indole, option B, pyridazine and oxazole, option C, pyrazine and quinaxalin, and option D, pyrimidine and thiazole. So, which heterocyclic rings are present in TPP or thiamine pyrophosphate? When you are reading the organic chemistry and also in drug development things, remembering the heterocyclic rings, the names of the heterocyclic rings and the classifications is very important, because organic chemistry is, most of the organic chemistry involves the heterocyclic rings, and when organic chemistry combines with the biology, or there are most of the things you will see there are heterocyclic rings. The 90% things are involving heterocyclic rings. So, remembering the names of heterocyclic is a very important thing for the organic chemistry and also for the biological studies. Okay. All of you try to answer this question, which heterocyclic rings are present in TPP or thiamine pyrophosphate? Option A is pyrazole and indole, option B is pyridazine and oxazole, option C is pyrazine and quinaxalin, option D is pyrimidine and thiazole. Is there any answer in the chat box? Let me check. So, option D is the most given answer. Now, let me check. So, the correct answer is very good, option D, pyrimidine and thiazole. So, this is thiamine is converted to thiamine pyrophosphate or TPP by TPP transferase enzyme, and ATP will be converted to AMP. So, here as you see, two phosphate groups are added, or the pyrophosphate moiety. There are two phosphate moiety which is together called pyrophosphate. So, these are added to thiamine by the enzyme TPP transferase. So, this is the structure of TPP or thiamine pyrophosphate. So, as you can see, there are two heterocyclic group. One is this with 6-member heterocyclic ring which has 2 nitrogen is 1 and 3 position, which is called the pyrimidine or pyrimidine. Okay. And there is a 5-member ring which has nitrogen and sulfur at 1 and 3 position, which is called the thiazole. Okay. So, the heterocyclic rings which are present in TPP are pyrimidine and thiazole. So, remembering the names of heterocyclics are very important. So, these are very important pharmaceutically, remembering heterocyclics. This is furan. Okay, sorry. This is furan. This is thiophene. This is pyrrole. So, 5-member rings, remember 5-member rings, there are if there is only one oxygen, it is called furan. If there is only one sulfur, it is called thiophene. If there is one nitrogen or NH, it is called pyrrole. If the 5-member ring has two hydrocyclic atom, based on this position their names changes. If there is oxygen and nitrogen combination and oxygen and nitrogen in 1-2 position, it is called isoxazole. If oxygen and nitrogen is in 1 and 3 position, it is called oxazole. And if there is nitrogen 1 and 3 position, it is called imidazole. Nitrogen and 1 and 2 position, it is called pyrazole. Sulfur nitrogen 1 and 2 position is called isothiazole, and sulfur and nitrogen is 1 and 3 position is called thiazole. Nitrogen in para position in 6-member ring, it is if there is only one nitrogen, it is called pyridine. If there is 2 nitrogen, so based on the position of nitrogen the names changes. Like if nitrogen is 1-3 position, it is called pyrimidine, and if it is in 1-2 position, it is called pyridazine, and if it is in 1-4 position, it is called pyrazine. And there is oxazole also, here it is the nitrogen 1-2 and oxygen in 3 position. There is isoquinoline, if there is 2 ring together, it is called isoquinoline, it is called quinoline, it is called benzofuran, and it is called benzimidazole. So, these are the names of few heterocyclic rings, and I would request all of you to remember the names of the heterocyclic rings. Okay. So, last question of today's session says that the biosynthesis of gamma aminobutyric acid or GABA from butyric acid involves number A, glutamate carboxylase as enzyme and biotin as cofactor, number B, glutamate decarboxylase as enzyme and TPP as cofactor, number C, glutamate decarboxylase as enzyme and PLP as cofactor, and number D, glutamate carboxylase as enzyme and PLP as cofactor. So, which of the following is correct? The biosynthesis of gamma aminobutyric acid from glutamic acid involves glutamate carboxylase as enzyme, biotin as cofactor, glutamate decarboxylase as enzyme and TPP as cofactor, glutamate carboxylase as enzyme and PLP as cofactor, and glutamate carboxylase as enzyme and PLP as cofactor. Let me check if there is any answer in chat box. Okay. Anyways, I will discuss the last question. We have already passed our time because we started very late today due to internet disruption at my site. Okay. So, the answer is number C. Glutamate decarboxylase as enzyme PLP as cofactor is involved in the biosynthesis of gamma aminobutyric acid from glutamic acid. So, what is gamma aminobutyric acid? So, it acts as an inhibitory synapse in the brain by binding to the specific transmembrane receptor in the plasma membrane of both pre and post synaptic neural processes. It is most highly concentrated in substantia nigra and globulus pallidus nuclei of basal ganglia followed by the hypothalamus and pericoccal grey matter. So, it is a major inhibitor and neurotransmitter in mammalian central nervous system, present in nerve terminal and released from electrically stimulated neurons. GABA functions by binding to specific transmembrane present in plasma membrane. So, this is the catalytic cycle. As you can see, there is a glutamate and it is converted to pyruvate, and there is a GABA formation here, and the enzyme which are involved is glutamate decarboxylase, okay, and PLP is the cofactor. So, you do not need to memorize this whole catalytic cycle, just remember what is GABA, what is the role of GABA and the name of this enzyme and PLP for the cofactor. Okay. So, this is it. This was the last question of today's session. So, today we discussed about your coenzymes, cofactor and also molecular biology. One question, there is a doubt about that sense and antisense strand question. Before uploading I will correct it. I think I did something mistake in that question. So, before uploading I will correct it surely. So, do not worry. So, today it was the week 6 live session. So, now I will share my email ID with you. If you have any doubt, any query regarding anything about today's class or anything, you can just let me know, and Bhavna, if you have anything to tell me, you can write in the chat box. Okay. So, this is my email ID. If you have any doubt, any query regarding today's class or anything, you can let me know by email. Okay. I will share this PPT and this video with you. There was many internet disruption and network power cut issue from my side. So, I thank you all for keep joining the class and do not for not leaving the class. So, this was the week 6 live session for the organic chemistry and biology and drug development, and yeah, thanks a lot. No, assignment you cannot submit through email, Bhavna. You have to submit the assignment for the NPTEL website. So, go to the SWAYAM website. When there is a assignment, you have to click the options there and you have to submit it itself. Do not email the answers, okay. Yeah, in the SWAYAM app, you have to in the SWAYAM app, you have to select the correct options and then you have to submit it there itself. You do not need to email me the answers, okay. You do not need to email me the assignment, you have to do it in the SWAYAM app itself. So, if you have any doubt, any query, feel free to reach me through the email, okay. In the next session, we will have on the next Tuesday from 6 p.m. to 8 p.m. So, I will close the session for today. Okay, so then you can actually write an email to the NPTEL course. There is a problem, there is a discussion forum, okay. So, you can just go there and let us know what is the problem, like what you did, okay, and I think you will get the answer from the email only, okay. Just go to the problem forum, the section, there is an email ID given in the SWAYAM portal. On that email ID, just tell what is the problem regarding the assignment submission and they will then answer what you have to do it, okay. I think they will consider, if you have emailed it, I think they will consider it, okay. So, you do not need to worry. From now on, please submit it on the website only, okay. So, thank you all. I will close the session for today. Let us meet for the next session on the next Tuesday.